Good morning, good evening, good afternoon to everyone. Welcome to the fresh episode on Drug Development Diaries. This is a YouTube channel that we are running, and we're discussing different aspects of drug development from discovery till production and till commercialization. We recently published a book called Approved, the Life Cycle of Drug Development. This came out somewhere in 2025. Springer Link published this book, and we invited a lot of experts as authors to write different chapters on drug development process. And in this series on YouTube, we are inviting these authors and we are asking them a lot of questions regarding how their chapters are structured, what are their thoughts on different trends in drug development, what are their career trajectories, and what kind of an advice they will give it to young researchers. So this is again another episode in this same series. I'm joined by our host, Dr. Naren Chirmule. Hello, sir. Welcome to the episode. And as guests on today's episode, we have Susan, Jenny, and Mariana. I would pass on now to Naren, sir, to introduce our guests on today's episode and get the episode going.
Yeah, thank you, Bihang, and welcome all of you to the episode. So I'm gonna, before I introduce Susan and Jenny and Mariana on the team on this podcast, I want to give a little bit of background on how I met Susan. And so I met Susan, I think in end of 2018 or 19 in Bangalore, when we were when she was in Thermo Fisher at that time, and we were talking about various things. I'd heard of her work for many years, but the one thing that struck me, you have to have a common theme, right? So she's a Mumbai girl, went to Xavier's College, and so I'm also from Mumbai, so that's immediately we connected, and so there's a lot of comparison. And since then we've been in touch for a long time. But Susan has more than 25 years as a molecular virologist, done seminal work in this area of cell and gene therapy. She's currently Chief Technical Officer and Chief Commercial Officer of Genzyme. She's got a lot of experience in this space, first from Brammer Bio, then Thermo Fisher, and many other companies in that. Like I told you, she went to undergrad in Xavier's College and then did her PhD in molecular virology, I understand, probably, right? Yes. Probably expand on that. Then we have Jenny. Jenny has a PhD also, from in the textbook. Actually, we have everybody's CV at the back, which is or bio. Interesting things that everybody has written in the textbook about themselves. Molecular biology and molecular genetics, and currently she leads the business operations at Genzyme. And Mariana, who is in Rio de Janeiro, is a postdoctoral fellow there at the Federal University, and has a lot of expertise in viral vectors, especially AAV vectors, and especially on this chapter that Susan has led. Mariana has done a PhD also from University of Göttingen in Germany, right? But maybe they'll speak about that specifically. So welcome to this podcast. I'm excited to talk to you about this. So, Susan, yeah. So my first question is a generic one, since gene therapy is sort of the talk of the town these days, and everybody's talking about this now. Can you explain to us, like, what is a viral vector? How do you deliver genes?
And I think Mariana is going to take that first question. Go ahead, Mariana.
Yes, sure. So viral vectors used for gene therapy, they are actually viruses that we turn them into delivery vehicles for therapeutic genes. So in gene therapy, we actually aim to deliver a genetic material to a cell with a therapeutic purpose. So to deliver this genetic material to the cells, we need some strategy that ensures that this material can enter the cells and that it will be expressed. So those are the gene therapy vectors that can be divided into viral or non-viral vectors. Viral vectors, they are very efficient as they take advantage of the natural ability of the viruses to infect the cells and deliver genetic material to them. So to build a viral vector, we remove all of the harmful sequences from the viruses, and we replace them with our therapeutic genetic material of interest for that application. And as a result of that, we have highly efficient vectors that also have a strong safety profile to use in humans. And viral vectors, they are actually now very successful, and they represent a large portion of ongoing clinical trials with gene therapy among the gene therapy vectors used, and they are also the majority of approved gene therapy projects on the market now. They also use viral vectors. So
Yeah, no, that's wonderful. Which are the recent viral vector-based gene therapies that have been approved that are also in the news?
Yeah, I think that's a very good question, Naren. And Mariana, if I may, I'll just jump in and answer this question. I think Sarepta's Duchenne muscular dystrophy product, which is Elevidys, was very recently approved over the last year or so. And that product is a adeno-associated viral vector that was serotype RH74, again, for the treatment of Duchenne muscular dystrophy. While we, really felicitate and are excited about that product and the way it has developed, there have been some, unfortunately, headwinds in that conversation as well. And definitely can't talk about any drug without actually talking about maybe some of the repercussions or some side effects associated with it. We have since then, gotten into a conversation about more immunogenicity and aspects of the viral vectors that we need to continue to solve. But those are definitely always in the foreground for any regulatory agency and all of the drug development companies that we partner with.
Yeah, because I think as exciting as this field is, as you mentioned, it's always a yin and yang. We'll always have to look out for what the
Correct.
adverse effects are, right? So moving on a little bit from gene therapies to gene editing, which now is the craze. Everybody from high school children know what CRISPR is, and it's amazing that the high school children are learning this technology. So from gene editing perspective, what's sort of exciting these days in gene editing? What's happening in this field?
Yeah, I can take that quickly. So as you mentioned, the CRISPR-Cas9 has kind of been the leading tool used in gene therapy or gene editing lately. Just last year there was big news in the I guess we'll call him Baby KJ with the treatment of the baby with a in vivo CRISPR therapy.
Yeah.
And that treatment, was first of its kind, breakthrough for the in vivo treatment. And the therapy, was developed in, I think, under 6 months or about 6 months. So it was a quick, personalized medicine that was provided.
Yeah, I think absolutely gene editing is something that I don't know, humans are taking becoming God, so to speak. But it's exciting that we can actually talk about the C word, right? Cure with therapies with gene editing. So speaking of future trends, what are the latest trends in sort of gene therapy and gene editing technologies that we anticipate coming?
So I was going to try to answer that, and Mariana, Jenny, please feel free to add on to that conversation as well. Naren, one theme that we're talking about here, whether we're talking about viral vectors or whether we're talking about gene editing, they're all advanced therapies. We're talking about not treating symptoms. We're talking about curing. The word cure, the C word, is like such a big deal. But we all know that we really haven't quite reached a cure yet. What we've done is we've been able to, at least at a genetic level, navigate some of the therapeutic aspects of these drugs to be able to either stall or decrease some of the symptoms to the point where you can then have the body get a good, a healthy copy of the gene to continue progression. So the earlier we can catch the conversation, the better off we are to limit any of the deleterious effects of that disease indication on those patients, right? So I think there is always going to be a constant equilibrium between what the standard of care is right now versus what these advanced therapeutics can bring to the table. So in that context, if you kind of take that conversation, we're talking more personalized medicine in a way we've never talked about personalized medicine before. Jenny brought up baby KJ. There are also various aspects of viral vector gene therapies that are patient foundation derived more than anything of N of 1 clinical trials, where now the agencies are allowing you to kind of go down this path and kind of bring this conversation to their very important in that conversation that I was part of was a parent foundation called Ellie's team. And the patient in this context was Ellie, who was suffering from a neurodegenerative disease. And being able to get her a viral vector AAV9 serotype-based product was really phenomenal in being able to cure or at least get her to a place where her epileptic seizures decreased, and was able to get to a conversation where they were able to replace one of a critical gene in her body that was a transcription factor that allowed to kind of break this conversation down. So if you take that conversation and you look at it, this is great because now you're kind of talking about therapies that are personalized, but also how does this then play a role as we think about it moving forward and being able to build this conversation out? And I think here is where novel conversations are happening, where tropism is more in the context of gene therapies, being able to get even more in the context of engineering capsids that are able to go further into this conversation, go very closely into the organ that you're trying to solve this conversation for, and not just dump a payload into the liver and have these deleterious effects we talked about, like we did with the Elevidys, right? So that's one aspect to think about it. Another aspect is one size doesn't fit all. We can't use the AAV9 conversation and be able to take a systemic conversation and move forward. We probably can do it ophthalmology. We can probably use it for neurology. We may or may not be able to use it for all kinds of systemic disease indications. DMD being one of them, which we've been in front of that conversation, but we need to be able to say, what does that payload look like? How much of the total dose can we be able to deliver? And can we move this conversation forward, right? The other aspects are taking the CRISPR toolbox and saying what other gene editing tools are available. Well, Cas9 was amazing, but there are the others that are more specific, more precise, base editing, prime editing, smaller Cas9 Cas variants that could be able to be in this conversation. Maybe they're Staphylococcus aureus based Cas9 or maybe Cas12. These are other conversations that we've had. Zinc finger nucleases continue to be in that conversation as well. And then so this conversation keeps going on. Non-viral vector-based products are also available. And so there is going to be an equilibrium between what the vectors, the viruses can do versus what the non-viral vector conversations can be. LNPs, for example, would be a great example of that. Using mRNA tools in concert with lipid nanoparticles might be another conversation to be had. And then to take all this and to think about more about gene in a box, gene in a computer, more AI-related conversations, more automaton, because there's so much data that has been collected over the years, ever since the Human Genome Project, that nobody has really truly been able to explore. And maybe there's more tools that we're not able to explore that are still there that we should be able to leverage.
Yeah, no, I think that's a great summary of many things that we anticipate coming, and almost all of these things, and they're moving so fast. But almost all of the things that you talked about are in your chapter, and so I'm going to hand it over to Vihang. But I want you guys to talk a little bit about what it took to get the graphics done. But over to Vihang for the next set of questions.
Yeah, this has been a really great conversation so far. So I would like to bring the audience's attention to the chapter that we all have written in the book, Approve the Life Cycle of Drug Development. So may I request all of you to just talk a little bit about how this chapter is structured, what are the different sections in it, and how different concepts are explained.
Sure. So the layout of the chapter really breaks down the gene therapies as we've described here. We give a brief overview of the history of gene therapy and the clinical and product approvals. But then we go into detail for each of the main viral vectors that are currently being used in clinical and commercial applications. You have an adenovirus section, AAV section, lenti and retro, as well as HSV. And for each of those sections, we go into the natural biology of the virus and then into the manufacturing of the therapeutic products and how, and then give an overview of the clinical applications of these products and some of the headwinds that, have been observed, as well as, some of the advantages of these products. And then we give a brief intro to the gene editing tools, as we mentioned, the CRISPR-Cas9 and the zinc finger nucleases, and some of the clinical applications for those. And finally, the chapter concludes with an analytical section, really going into kind of how we approach quality control of the viral vector manufacturing, analytical methods that are used commonly to ensure safety, efficacy, and quality, as presented for each of the vector types. And then finally, it concludes with, just an overview, as a little bit of what Susan was mentioning with, kind of the outlook in the future for the gene therapy field.
When Naren first approached us to write this book chapter, he reached out to me. And my first answer was, no, there's no way. This is just too much over our day jobs that there's no way we can do it. And Naren walked me through it. He's like, I'm going to support you 100%. This will be a lot of fun. And to be honest, it was a lot of fun. And the way we approached this was we identified people that were interested, scientists that were interested to learn more or do more or explore or expand their repertoire of knowledge in this conversation, and maybe open doors for them in viral vector gene therapy. So I will definitely encourage other scientists who really don't think that they can do this to, learn how to eat an elephant one piece at a time. And was literally that. Eating this elephant was one piece at a time. Mariana was phenomenal in working on aspects of adenovirus and AAV. Sayuri Kato wrote the HSV stuff. That was her way to get into understanding gene therapy better after taking a break to raise kids. And I want to just call out, as women, it's always important to remind ourselves that our reproductive years are also our years when our career is really taking off. And to be able to come back into the field after taking a break was phenomenal for Sayuri. And bringing Sayuri onto this conversation was phenomenal as well. She did a phenomenal job with the HSV components. We also had Jenny jumping into lenti and retroviruses and really focusing ourselves on this is what we do for a living in our CDMO world. And she did a phenomenal job kind of getting that all buttoned in together and then focusing on gene editing as well. And then we had other authors that came in that helped to write.
Shashwati and Srinivas, yeah.
Srinivas did a great job with the gene editing, and then we tailored it a little bit as well. And then Shashwati really brought all the analytics together for us. So in like piecemeal, we were able to kind of build this book chapter. Naren had said, keep it simple, keep it small, and then it just became this giant kind of conversation. I think we were one of the bigger book chapters, but it was so much fun to bring it home together.
Yeah, and it is a book. It is a book in of itself.
Yes.
Yeah.
And the title itself, Hope Unlocked. That was Naren, your idea, and it was phenomenal because that was truly what we're doing. We're unlocking hope here, and it was really a great idea, and we all love the title there as well.
I also want to use this opportunity to call out Kitki and Ria, who made the graphics for you guys, and Jenny. The number of edits that we all had to go through for the it was phenomenal. It was a phenomenal piece of collaboration.
And I still use those graphics. We do some guest lectures through Jenizen, teaching manufacturing at some of the local universities, Ohio State University being one of them. And we have definitely generously borrowed from those graphics. So thanks to Kate Key and the team for helping us to put those together as well.
Awesome, awesome. Could you all talk a little bit about your personal career journeys, how you ended up in this profession, what all steps you have had, how has your journey been, and then maybe comment a little bit on what advice would you give to young researchers or students and young professionals who want to pursue career in this particular gene editing, gene technologies, and such fields.
Mariana, you want to tee us off?
Yeah, I can start. I am an early career academic researcher in Brazil. I'm a postdoctoral fellow, and I actually work. I do research in the area of gene therapy for more than 10 years. I started still during undergraduation when I did an exchange program, and I worked in the gene therapy center in the University of North Carolina. So since my undergraduation, I already had contact and I knew that I wanted to work with gene therapy. And then I came back to Brazil and I started my master's degree. And I also worked with gene therapy and AAV vectors in the context of retinal neurodegeneration. And I moved to my PhD, where I did a joint PhD here in Brazil and in the University of Göttingen in Germany. And it was the same thing. I was still working with AAV, so I have been working with AAVs for quite a while. And now I work with new experimental gene therapies in the context of neurodegeneration. So that's kind of my career path so far.
What's your advice to the students that are reading this book chapter?
Wow, I don't know. I think that gene therapy is a very new research, is a very new field, is a very new therapeutic approach with a great potential. So I would advise them to embrace this new technology, and really, because it really has a lot of potential to make a great impact in human health.
Jenny, you want to go next?
Sure. So I started out on kind of the classical academic path. I earned my PhD in microbiology and immunology, and my focus was mainly in bacteriology, molecular bacteriology. And I did several years of a postdoc research and then realized I wanted to, kind of get into more of application and industry. So Susan was an integral part of my transition into the industry. She was at Bremer Bio at the time, and I reached out to her, and she was able to help me enter the quality control team at Bremer Bio, which was a viral vector manufacturer. And I worked in quality control there for several years. And then as I was working there and learning more about the viral vector CDMO world, I realized I wanted to be on the commercial side and help to kind of use my scientific background to help bridge that commercial and technical operations part of things for the new clients. So I moved over to the commercial operations team after Bremer was acquired by Thermo Fisher, and supported the proposal and program design, and continued there for several years before, again, Susan was one of the key factors in helping me progress my career along, and moved over to Genesen. And now I lead the business operations team, where I still am, overseeing that program design, proposal design team, as well as kind of working with supporting the business development team and more of the technical aspects, and helping to bridge some of that between that technical and sales side. So my career for new students and emerging people coming into the field, along with the fact that go ahead and pursue these new technologies, and, keep reading the literature and staying up to date, but also keep those networks open and don't be afraid to take the open doors. Sometimes I know in my own career, I've been hesitant to make changes and transitions, but, if it weren't for the mentors and advisors around me encouraging me to continue to grow, I wouldn't have ended up where I am and, being successful and developing in this career.
Thank you, Jenny. And I will close out the conversation with my career, which I call a career of serendipity, because I think I just walked into conversations that felt like would be the best way to think about it. I really didn't have a plan on where I wanted to be and where I wanted to go, except to say I'm a medical school reject. And so that's my first advice. Rejection is good because it sets you on a path that takes you differently and sometimes more passionately towards where you should be, where your goal should be. I joined Xavier's and did my bachelor's in microbiology and absolutely hated it. I lived in the suburbs in Mulund, in the northern suburbs of Mumbai, and traveled to town to Xavier's. I hated looking through a monocular microscope and looking at these. I probably broke more slides than anybody else in the team there, like trying to do a gram stain and all of the other stainings. And then, really walked into doing a PhD in insect virology, of all things, in a small town in Lubbock, Texas, at Texas Tech University, primarily because that was the best path for my mom, my parents, to allow me to come to the US, since I was only 22 and the first child and the first girl child to leave my family and come abroad. So I had family there. My mom had had some relatives there that actually allowed me to think about it in that context. And insect virology itself was boring when I was studying microbiology, but insect virology on top of that didn't seem like quite the place I wanted to be. But it is the serendipity that actually brought me to viral vectors. And, long story short, as I navigated my career through a postdoc and came to the University of Florida, and as I was looking for what I wanted to do next, I realized that tenure track professor wasn't the path I wanted to go after all. After spending a significant amount of my career, half my career has been in academics, I realized that was really what got me into viral vector gene therapies, because the founder of Brahma Bio initially was Florida Biologics, was a professor, Dr. Richard Snyder, who worked in the same team as me at the University of Florida. And I reached out to him looking for a job. And when he realized I worked with insect viruses, when he realized I had baculovirus experience, that's really what was the cinch that put me in this conversation. And he pulled me into, made a role for me at Brahma Bio, Florida Biologics, to be able to kind of talk through process development. And then at the end of that, there was no end to that conversation. I just flourished in this conversation. I figured out this was my passion. This is what really makes me tick. Drug development and being able to work with viral vectors is really what helped me to stay focused. So in concert to what Jenny said, I would say, get out of your comfort zone. Ask the questions. You know all the answers to all the questions you don't ask or know. So rather than get a no for something that you've pre-initiated in your mind, just ask the questions. Stay curious. The network is absolutely critical. Surround yourself with people who will challenge you, who will push you outside that comfort zone, because you've got to be comfortable being uncomfortable. And then you will find what really puts that fire in your belly. And finding that fire in your belly is phenomenal, because you wake up every morning going to work and it's not work anymore. It's actually fun because you're really passionate about this. And I'll end with one last comment, and that's courage of conviction. Asking and challenging are critical things. Most of us, especially women, and especially immigrant women in this country, are going to struggle with asking those questions. So lean in, get a place at the table, and ask questions that will force you to have the courage of conviction.
Oh, that was amazing. Thank you. Thank you, all of you, for sharing such fantastic experiences. This is precisely what we want this podcast to portray. So Thank you very much for an amazing session. I mean, I think we talked about a lot of things. I know we could spend hours talking about everything in the book, but it's an exciting chapter that you've written. I've read it multiple times, and I also use your figures, by the way, for my talk. Sometimes they're so phenomenal.
Great.
Yeah. So I'll hand it over to Vihan to sort of close this session for us.
Before you close, can I just say a huge thank you to you, Naren, for allowing us to be part of this conversation. It's been really fun, and it's really brought us closer to Mariana, who we did not know until we started writing this book chapter. So it's been really great to be introduced to Mariana and getting Sayuri in the conversation as well. So thank you.
Thank you. Thank you.
Thank you so much. This was a great, insightful discussion. Susan, ma'am, when you said that, rejection sets you on a path, I could absolutely find it relatable. Your experiences of growing up in Mumbai, even I grew up in Mumbai, and I know what the perils are of traveling in a local train early in the morning from northern suburbs to southern areas of the city. So yeah, definitely, it was great learning about your career, your experiences. To Jenny and Mariana, also, it was great to hear about your paths and your advices to the students. Totally appreciate this discussion. So we are closing for the episode for today. To all the viewers, I hope you have learned something out of this. If you have any questions, let us know in the comments. Tell us what you think about the book. Tell us what you think about gene therapy, gene editing, and different trends in this part. If you want to learn something more on these lines, let us know about that as well in the comments. We will be back again on this particular podcast with different guests who are authors of other chapters, and we will be discussing different aspects of drug development in upcoming episodes of this series. Closing on, thank you again for all the guests. Thank you, Naren sir. To all the viewers, stay tuned for the next episode. Stay inspired, stay curious. See you in the next episode. Bye-bye.
Bye.